Hello, my name is Russ Dashtaki. I'm a biological sciences tutor here at Winston-Salem State University. You can find me on the first floor of Hill Hall. If you need any help, please come see me. Today's video is on transcription and translation. Transcription and translation are very important for the cell. Transcription is the process of taking DNA and copying it into RNA. Translation is the process of taking RNA and making a protein. The protein could be things like hemoglobin, could be some kind of enzyme like saccharate amylase, which is in your mouth, in your saliva, that breaks down carbohydrates the minute you put a piece of bread or French fry or potatoes in your mouth. Complex carbohydrates take a while to break down. So you need enzymes for that. So proteins can be enzymes, can be, muscle, just like when you eat meat. You're eating skeletal muscle from a cow, chicken, things like that. But it can also be something small, like hemoglobin, which carries oxygen. As long as you have iron, you make that protein. That hemoglobin can carry 4 molecules of oxygen on a red blood cell. So you've got so many of those that bring the oxygen from your lungs to the cells in the mitochondria to make energy. So we did a video on that earlier. If you want to see that, you can look at my cellular respiration video. Now, DNA is double-stranded, okay? The word for that is deoxyribonucleic acid. DNA, as you can see here, is double-stranded. That double-stranded DNA is very stable, and it stays in the nucleus. The nucleus is like the vault or the fortress where you'll find that DNA. Now, your DNA is like your cookbook. Let's say your grandma gave you this cookbook and it's, 100 years old. And it's been handed down through the family, and now you have it. Well, you find this recipe for this cake that's really good that you remember your grandma making, and you're like, I want to make this cake. So what you're going to do, you're going to take your cookbook off the bookshelf. Now, if you take it to the kitchen, you can get flour, grease, oil, egg on it, and you can mess it up. You don't want to do that. So what you're going to do is take that cookbook, find that recipe for that cake, and go to a copy machine and copy that recipe. Then you put the cookbook back on the bookshelf where it stays safe, and you bring that piece of paper that you copied, and you bring that copied recipe to the kitchen. And then you make your cake. If you get oil, grease, flour, egg, milk on that piece of paper, you can just ball it up and throw it away. So it doesn't matter because you've got the original cookbook in your bookshelf. Nice and clean and safe. Same here, okay? So your DNA stays in the nucleus, okay? That's like a vault in there, okay? It protects that DNA. Well, let's say you want to make hemoglobin, that protein I was talking about that carries oxygen on your red blood cells. Well, in order to make that, you need the recipe for it. So you find the recipe for making hemoglobin on this red blood cell. And then you take that recipe and you copy it. You're like, okay, I want to make this hemoglobin to put on a red blood cell. So you find the DNA, you find the recipe, and you copy it. The process of copying that from DNA into what they call messenger RNA, which is like your piece of paper that you copy from the copy machine. That process is transcription, transcribing. You're copying a section or a piece of that DNA into RNA. And you're doing that in the nucleus of the cell. That's important to understand. Transcription occurs in the nucleus of the cell. And then, once you transcribe that DNA into a piece of RNA, just like you copied the recipe on the copy machine, then you take that copy of that piece of paper to the kitchen. In this case, your copy is that RNA, that single-stranded ribonucleic acid. Remember, DNA is double-stranded, deoxyribonucleic acid. The base pairs, again, we talked about, are A, T, G, and C. the base pairs of RNA are A, U, G, and C. So there's a uracil instead of the thymine. So adenine, uracil, guanine, and cytosine. So you make that copy. You transcribe that DNA into RNA. And then you take it to your kitchen. So you leave the nucleus, just like you took that piece of paper from the copy machine and took it to the kitchen to make your cake. You take that RNA, that single-stranded RNA, single-strand, see that? You take that single-stranded RNA and you take it to the kitchen. Well, what's your kitchen? It's the ribosome. Remember, my name is Russ. Russ says ribosomes make proteins. Russ says ribosomes are on the rough endoplasmic reticulum. Ribosomes make the rough endoplasmic reticulum rough. So you can remember that. All R's. Okay? So, the kitchen is going to be the ribosome. It's either free floating in the cytoplasm, it makes proteins there, or it's attached to the rough endoplasmic reticulum. Ribosomes are what make the rough ER rough. So you find a ribosome and you take your RNA to that ribosome. And that ribosome will copy that RNA into a protein. That process is called translation. Copying a piece of RNA into a protein is called translation. So here we have our recipe that we copied, okay? And we're taking it to our kitchen, the ribosome, to make our cake, the protein, okay? So if you think of it that way, hopefully that will help you remember the difference between transcription and translation. And again, if you need anything, I'm located in the first floor of Hill Hall. I'd be glad to help you. Again, my name is Russ Daishaki, and I'm a biological sciences tutor. And I hope you have a great day.